Hello and welcome to this quick tour through MetOSite, an integrated resource for the study of methionine sulfoxidation and its biological implications. After launching your favorite browser, you must type the following URL: metosite.uma.es. In this way, you will reach the resource we have called MetOSite. At the heart of MetOSite, there's a large database of manually curated data related to experimentally confirmed sulfoxidation sites and the effect of such post-translational modification on the protein's properties. This database can be reached programmatically using our API that we'll show later on, or just browsing the web app. If you opt for the second possibility, you'll find 4 tabs: Species, Oxidant, Scan, and Search. The first 2, Species and Oxidants, simply offer a summary about the data you will be able to find in MetOSite. This information is presented putting the focus either on the species or on the oxidant. For instance, if we click on the Species tab, you will be able to find a table where you can see what species are represented in the database, as well as how many protein and how many methionine sites each species contributes to the database. Alternatively, if you click on the Oxidants tab, you'll find a table that collects the number of methionine sites that have been described as methionine sulfoxide after treatment with a particular oxidant or stimulus. More useful and interesting are the following 2 tabs: Scan and Search. Let's start describing Scan. This option is indicated when the user is interested in a specific protein and they want to find out if any of the methionine residues of this protein have been detected in previous works at Meto. Let's say we're interested in the cytochrome C. Then, if we know the UniProt ID for this protein, all we have to do is introduce it and click Scan. On the other hand, if we do not know the ID for the protein of interest, we can type the name of the protein and click Scan. The server will show us a list of all the proteins found in our database whose names contain the typed word. If the protein we are interested in is in that list, all we have to do is click on the shown UniProt ID. This action will lead us to the report for that protein. This report includes general information about the protein, specific information regarding the modifiable methionine residues, and, whenever possible, information related to other PTM different to sulfoxidation. On the other hand, if you're interested in a set of oxidable methionines rather than a concrete protein, the place to go is Search. Once at the right place, we have to choose the setting for our search by following the 5 steps we describe next. First, we have to choose what group or groups we're interested in. For this purpose, the user must be aware that each methionine found in our database is assigned to one of the 3 possible groups. Group 1 includes all those methionine sites coming from high-throughput studies, and nothing is known about the effect of their sulfoxidation, just because it has not been addressed. Group 2 is for those residues for which the potential effect has been investigated, but no effect could be found. Group 3 is formed for those methionine sites whose sulfoxidation have been reported to have an effect on at least one biological property. Herein, we have selected Group 3. The next step is to filter results on the base of the biological property affected by the sulfoxidation. To this respect, we have considered 6 properties: gain in protein activity, loss in protein activity, gain of protein-protein interaction, loss of protein-protein interaction, effect on protein stability, and effect on subcellular localization. For each of these properties, we can mark yes, meaning that such property has been observed for all the methionine sites we intend to recover from the database. On the contrary, if we mark no, it only means that we haven't found experimental evidence to support such an effect. A different alternative is to select either. In that case, it means that you do not care about the property, so the recovered sites may or may not affect that property. For the sake of concretion, suppose we are only interested on those sites that, when oxidized, lead to an activation of the protein, but we do not care about the other properties. In that case, our choice would be the one shown on the screen. Next, in step 3, we have the chance to filter by organism. Following with our example, let's say we are only interested in bacteria. Then, in step 4, we can filter by oxidant. In our example, we're going to choose all of the oxidant. In other words, we do not want to filter. In the last step, all we have to do is send the query and wait for the results. Once we have the search results, we are offered the opportunity to learn more about the recovered protein through the link to a UniProt, or by just going directly to the scan report for this protein through the provided link. With respect to the API, they provide 8 endpoint functions grouped into 3 categories: proteins, sites, and summaries. We invite the interested user to explore them with the help provided in the document section, where you will also be able to find an outline for the API. We hope this resource may be of any help to you, and if you have any questions regarding MetOSite, don't hesitate to contact us.